Hello, everyone, and thank you so much for joining us today for another one of our webinar series from the IPDGC, where we have talks revolving around neurodegenerative diseases and Parkinson's from experts in the field. My name is Natasha. I'm a PhD student at the University of Luxembourg, where I study Parkinson's disease, and I have the pleasure of moderating these sessions. Today, we are really excited to have Dr. Mohamed Salama. So he received his medical degree from Mansoura University in Egypt, and he established the first Translational Neuroscience Unit in Egypt. Today, he's going to be telling us a bit more about the establishment of the Egyptian Network for Neurodegenerative Diseases, its research journey, and the future goals of this network. So with that, I'm going to hand over to Dr. Salama, and we're really looking forward to hearing about the work you've been doing. So yeah, the floor is yours.
Okay, thank you so much, Natasha. Thanks for IBDGC for giving me this nice opportunity. Certainly, I'm happy to present my story, my journey. When I was invited in the first place, I was thinking of maybe sharing some research projects, maybe talking about one of scientific points. However, I thought it would be much more helpful if I shared a nice story with you. The story of establishment of the Egyptian Network for Neurodegenerative Disease, because simply inside this story, we could find several concepts that is highly related to starting research in the field of neuroscience or in general, we are generally speaking, in Africa. How to face problems, how to tackle different issues. So in brief, I sorted out that telling the story of ENND would be much more appropriate to our situation today. So in brief, my name is Mohamed Salama. Currently, I'm associate professor at the Institute of Global Health and Human Ecology at the American University in Cairo. I'm telling you the story, as I said, of ENND. In principle, we'll be passing through an introduction of translational neuroscience and translational research in general, ENND stories, and some facts maybe you could receive as achievements. I don't know, but they are facts. Some milestones achieved by ENND, and finally some opportunities that I can see in the area of neuroscience and research inside Africa. First, I would like to introduce the concept of translational research because it's crucial from my point of view. And this is one of the points I started to fight since my return to Egypt, trying to establish. What's translational research? Is it important or valid or valuable to start translational research in any place, especially in low and mid-income countries? So first, let's know or understand what's meant by translational research. Usually, when we try to speak or talk about translational research, most of scientists focus on the first part of the process, which is certainly very important, which is crossing or bridging the gap between basic science and clinical practice, which is important. Yes, certainly, this is a very important part of translational research. However, most of the time we forget to talk about the second stage, which is adoption of the best practices, adoption of the final product to be used by the community. In other words, we have two unique compartments for translational research. Both are complementary to each other. The final output, the final goal is to help the community, to help patients, to help our population. So to reach this goal, we have to pass through those two processes. I remember a few years ago, I was talking about translational research in Trinity College, and one of my colleagues there in the Global Brain Health Institute told me, Mohammed, you have been talking a lot about the first definition of translational research. You ignored or neglected the second part, and that's true. Now I'm trying to complement both parts. So first, as I mentioned, translational research, T1, or the first stage of translational research, is to cross or bridge the gap between basic science and the clinical practice. The second is the adoption of the solution. Okay. according to previous definition, we could have two types, as I said, T1 and T2. For this definition, it's a very nice one, and by the way, it's saying that it is a unidirectional continuum, and this is true. It's continuum. It's unidirectional also in reaching the goal. So as we said, the final goal would be helping the community. The final goal would be decreasing or improving the condition and decreasing the pain or the bad situation or the suffering for patients. However, this is for the goal. Sometimes it could be bidirectional. I mean, when you read this definition, you feel that, okay, it's only unidirectional. We should start from the lab, working on cell culture or working on animal models, and then move forward to human or clinical trials. That's true. This is one of the directions. However, there is no problem, no worries to go back sometimes. Sometimes we have clinical observation, clinical findings, some findings from human trials. We have to confirm and validate. In this case, you have to go backward. So that's why I insist to say that it is sometimes bidirectional, especially in the first stage. So in the first stage, yeah, most of the time we walk from basic research to human trials or clinical trials. However, sometimes we need to go back and confirm our findings. However, the final output would be certainly in one direction, which is adoption of the solutions for improvement of condition of patients, population, our community. This is a very nice diagram I usually use, which is representing crossing the value of this. Why it is called the value of this? Why we are calling that this translational gap is called the value of this? It is true because it witnessed the death of many of nice and innovative ideas in this place. So usually you, as a basic scientist, you have bright minds and you come up with idea, then start hard working on this idea in your lab and finalize and publish a paper, and that's it. For me, to finish with publishing and the paper, and that's it, is simply the death of an idea, because we need this paper or this idea to cross this area and go there to the clinical trial, clinical application, and finally adoption for solution. If you need to estimate or evaluate the size of this gap, simply go to PubMed and write on your keyboard the words Parkinson disease and neuroprotective agents. You'll find thousands, maybe hundreds of thousands of nice articles published everywhere talking about neuroprotective agents that have been claimed to cure Parkinson disease. And most of the papers are published talking about basic research. So scientists and basic scientists are working in their labs, closing their labs, and start working hardly on cell culture, on animal models, whatever, and come up with final results and publish them. However, try to go to PubMed again and cite and write Parkinson disease neuroprotective agents in humans. You'll find great decrease, major decrease in the number of papers published working with neuroprotective agents in humans. You need to see how the gap is big, go further and search in clinicaltrials.gov. Search on new drugs for treatment of Parkinson's disease adopted by physicians now. You'll find one, two. So we started with thousands or even hundreds of thousands of ideas and basic research, ending up with one or two drugs that may be adopted. So the gap is increasing, the gap is enlarging. And if we couldn't find some solution for this, it would be catastrophic. That's why I think two great scientists, both Elias Zeroni and Rudi Belling, both noticed this enlargement of the problem, the translational problem. And according to their perspective or point of view, the cause of this large gap may be that biologists haven't a clue about real medical needs. Maybe clinicians are deeply involved in their clinics. The major problem is that we have two teams. We have two worlds, the world of basic science and the world of clinical practice. A basic scientist has been working in his own lab for years, but certainly he doesn't understand 100% the needs of a clinician. For example, I, as a basic scientist, I would go to the lab and work on pathways and signaling and molecules related to neurodegenerative disease. However, on the other hand, the clinician would be more interested, more keen to find a solution for improving the quality of life. And simply, if we can't get a treatment, a cure for the disease, it's not the end of the world. At least we try to improve the condition, to decrease suffering. So from a clinician point of view, improving the quality of life, the value, the disability may be equivalent to having a cure for the disease. Sometimes I could use a symptomatic treatment that improves the conditions and the patient is controlling his manifestation. That's fine for me. On the other hand, the basic sciences feel that no, we are trying to cure the pathology. If I don't have a treatment for such a pathology, then I didn't make an improvement. And both sides are mistaken. Both sides are working together alone, and this is a major problem in our current world. I also called this very nice title. It's a point of debate and lecture in Australia, I guess, in the beginning of this century. And the title was, "Can we afford to fund translational research?" However, the lecturer ended up saying that actually the question should be, can we afford not to fund translational research? And that's true, 100% true. Not even in low income country, but also in rich countries. The richest countries have limited funds. Funds are limited. So we have to focus our research on those who had a goal, of those who can really offer solutions at the end of the day to the community and population. For that, funding translational research is a must. And actually, by talking about funding translational research, we do not mean that we should stop funding basic research. No, certainly this is not a solution. However, it should be basic research with an eye on possible applications. Funding basic research with plans how to improve if the successful grant goes on and you find or reach it valid results, how to improve, what are your plans? So simply, we are thinking of the steps further. So I'm not saying that we should focus only on clinical trials. No, certainly this is not a solution. But we have to start looking for multidisciplinary team. We have to look for a team composed of basic scientists, team composed of clinicians, team composed of geneticists, all working together to find solutions for different diseases, for different problems in the community. And to show the importance of translational accomplishments, Society for Neuroscience published this all publication actually talking about translational neuroscience accomplishments. And I showed you here the table of contents. So if you can see how many disorders or problems that have been improved based on translational approaches. One of them here is our area of interest here, which is Parkinson's disease. Do you know what is the translational approach that offered the highest impact in Parkinson's disease research? Animal models. So this is very important. When we start talking about translational research, you might think of, okay, we are talking about genetics. We are talking about transgenic approach, gene therapy, whatever. No, actually, the major translational accomplishment in Parkinson's disease was developing animal models, and this can be certainly understood because developing a realistic animal model for Parkinson's disease will enable us to start understanding the pathogenesis of the disease, understanding the possible pathways of the disease, and trying and evaluating several neuroprotective agents before moving forward to the clinical trial stage. So yes, animal models is considered a translational approach. Although it seems like basic research, however, it is sort of translational research. With that in mind, we started the dialogue initiative. So I returned after finishing my work in Philipps University Marburg in Germany, and we were thinking of new vision, having a multidisciplinary team working in the area of neuroscience. This was in 2011, by the way, and the dialogue started by that time, and we faced the usual problems. So for physicians, for scientists like PhDs, most of the time when you are talking about collaboration with clinician, you will be faced by this. Okay, those clinicians are very superficial. They don't think about the amazing world of signals and molecules and they are just looking for their trials, ending up with publishing in a clinical journal. And actually, this was a feedback received by one of my colleagues and in Europe. So yeah, there is somehow a misunderstanding. On the other hand, when you start talking to clinicians, they are very busy. They say, Mohammed, it's not the time to start thinking about those guys in their closed labs. We are working in patient in real life. We know the real life situation. So please don't start disturbing us, calling for collaboration with those guys talking all the time about molecules, signaling, and so on. So we started the dialogue. We had several problems. However, we succeeded eventually after 2 years to establish the first multidisciplinary group for studying neurodegeneration. This is the meeting where we launched the network, and we named it Egyptian Network for Neurodegenerative Disease. And as you can see, it started as a collaboration between 14 Egyptian universities. This is the geographic distribution of the universities. And again, as you can see from the map, I don't know if it is clear or not, but it is covering all over Egypt. So actually, when we start thinking about recruitment of patients or cases or whatever, it's a collaboration between literally most of the cities of Egypt, most of universities and 5 cities of Egypt. We have centers in Cairo, we have centers in Upper Egypt, we have centers in Delta, we have centers in North Coast, we have centers in South Canal, and so on. So this was the first accomplishment, to have the network established, and we succeeded. We put the aims, and again, we have the main aim is to promote research in the field of neurodegenerative disease. Also, we had another aim to exchange knowledge and experience in neuroscience, guided by the collaboration with different countries like Europe and US, UK, and so on. To foster education, Egyptian Network for Neurodegenerative Disease, I think so far have organized around 10 international meetings with collaboration with IBRO, ISN, Movements Order, and more. And in those workshops or summer schools, we hosted students as well as international faculty that helped to increase the awareness both of neuroscientists and neurologists of the value of multidisciplinary research. And the most important aim was to further the dialogue between basic and clinical science. If we went back to this photo, you will find that the team is composed of different partners. Some of them are clinical neurologists, some of them are basic neuroscientists, some of them are geneticists. Actually, some of them are artificial intelligence and machine learning experts. So I could claim that ENND represents a multidisciplinary network. Okay, next step was we need to put our goals. We need to start thinking, how are we going to approach the problems of neurodegenerative disease, especially Parkinson disease, in our place. And with that said, I remember a nice remark given by Rudy Belling. Rudy Belling is the founding director, I guess, of Luxembourg Center of System Biomedicine, and he visited Mansoura a few years ago, and then started discussion with scientists there. And when we had dinner by that time, he told me, Mohammed, you are missing the most important point. Yes, you can do research. However, if you need to do research, competitive research, you have to think about your strengths and your potentials and capacities. So please don't talk to me about stem cells and high technology and in a lab in Africa, for example, when you are trying to compete in a lab in the US or Europe. However, think about some approaches like studying your environment, studying your genes, studying your patients. This could be valid. This could be very competitive when you are applying for a grant or applying for application and so on. So we put this in our eyes and started thinking what experts say, what should be our goals. So we have several answers. One of them is very important. This is an outlook by nature about Parkinson's disease. And as you can see, they identified 4 big questions to answer. And if we answer, we could get the big lottery ticket. So first, how does Parkinson's disease begin? And as you can see, I highlighted what should we do? What are the next steps? Because something have been done, but we, as scientists, we have to move forward. So the next step in understanding how Parkinson's disease starts is to search for blood and urine-based biomarkers. Okay, we need biomarkers to early diagnose or predict the disease before clinical manifestation. Great. So what is this question again? It's question 4. What is the best way to divide people? Important, magnificent question. How to stratify patients? Very important, because simply stratification and classification of patient is the key for precision medicine. Simply, we are now in a time we cannot give one tablet or one capsule for all types of patients. We have to understand that even if you are diagnosed with Parkinson's disease and your neighbor is Parkinson's disease, you might have 2 different types of Parkinson's disease. How could we classify patients? The first would be genetic approach. So genetic research could provide some clues to understand the differences between different patients, different subtypes of patients within the major category or the general category, which is Parkinson's disease. So with that said, we defined 2 major goals for ENND in the starting 5 or 6 years. We have to tackle genetic studies and biomarkers. As I said, genetic studies were very important to understand the patterns of the disease and to start stratifying our patients. Biomarkers, as I will mention later on, are very important, especially in neurodegenerative disease, because simply when Parkinson patient comes to your clinic with his hand shaking, with some bradykinesia, with some clinical complaints, this means that the patient has already lost about 85% of his neurons. So simply, it could be useless. Yeah, usually you are trying to save something that's already lost. Can you imagine what could be happen if you can diagnose or identify the patient much earlier when he just lost 20% of his neurons? The change could be amazing. And that's why we managed to put those goals as ENND translational goals, genetics and biomarkers. Okay. So let's discuss the first approach, which is genetics. As you might know, I know that you are all experts in Parkinson's disease, or I hope. We have 2 types of Parkinson's disease from a genetic point of view, idiopathic and familial. For idiopathic, we mean that simply we don't know the cause. We don't know the main trigger. Certainly, genetics play a role. However, it's not only genetics. And that's why usually we call it is an environment-gene interaction. It's not just genetics. You might have the risk SNP, but you don't have the disease. That's why it's called idiopathic Parkinson's disease. On the other hand, we have familial Parkinson's disease. In familial Parkinson's disease, the inheritance of the disease is following a simple Mendelian inheritance model. So simply, if you have the mutation in this gene, you will get the disease. Idiopathic is difficult, very difficult to study. Why? Because as we said, risks are variable, and you can't identify one single risk. So it's genetics, it's environments, it's lifestyle, it's a lot of variables. So it's very difficult to study and very difficult to correlate. However, idiopathic Parkinson's disease constitutes 85 or 80% of Parkinson's disease. So this is the majority of the disease. You can't ignore, even if it is difficult. For familial, the number is much low. The percentage is much low. Yes, it constitutes around 10% of Parkinson's disease. However, it's very important here because simply you can identify with certainty that this gene is responsible for Parkinson's disease. And once you identified a responsible mutation or gene for Parkinson's disease, you simply can say that this pathway contributes to the pathogenesis. So although you are here studying the minority, but you can come up with theories for the pathogenesis of Parkinson's disease in general. So let's say both are appealing, both are important to study. We started with the idiopathic, and this was the first project for ENND. We established our network of neurologists and scientists. We launched our SOPs. We designed and established our recruitment strategy. I claim that we established the first biobank for neurodegenerative disease in Egypt. We also started the capacity building program through sending our junior scientists to Europe and Germany and receiving international faculty start discussing with us the potential for improvement and training. And finally, the end result is we reached 500 idiopathic Parkinson's disease and similar number of matched controls. The value actually is not with this number of cases. And when you are talking about 16 or 14 recruitment center, when we are talking about population like Egypt was 100 million, you could say that the number is much low. And I agree, yes, it's a very low number. But the lessons learned from the first experience is amazing. And actually, we learned a lot of problems. We learned from many mistakes that each center contributed. Yes, we gathered much more patients at the first time, but we noticed that, okay, we could have some samples without related data. We could have some samples without accurate data collection or clinical data or environmental data and so on. So I could say that those 500 samples may not represent 100% of the study. However, they were a very important net result based on the fact that this was the first national study in Egypt trying to collect and recruit Parkinson patients from all over Egyptian population. This was highlighted earlier in Nature Middle East, and then later on, this was the result. The results were published in scientific reports, and I would say that the results were not what we expected previously. One of the also mistakes we have done that we dealt with the Egyptian population with European standards. So we thought, okay, so we are going to screen Egyptian patients with the known genes responsible for risk or the risk genes in Parkinson patients. So we screened them for a panel of around 80 SNPs, expecting that from a well-known risk genes for Parkinson's disease, we could come up with, let's say, 75% of positives. So we identify many risk SNPs or replicate confirm the results in the Egyptian population. We forgot a very important point, in fact, that Africa, and Egypt is part of, is somehow different, especially in the makeup, the genetic makeup, the environmental factors, and more. We are totally different. So from the 80 SNPs we screened, we only identified one SNP, which is related to detoxification of organophosphate type of pesticide. The value of this SNP, actually, it is an interactive SNP. We called it an interactive SNP because simply this SNP increased the risk of Parkinson's disease only in condition of exposure to the risk factor, which is pesticide. So if you are exposed to pesticide and you are carrying this variant, you have increased tendency to develop Parkinson's disease. If you don't have the exposure, you are not exposed to the risk, most probably you won't get the disease. So this is a very good model for gene environment interaction. In addition, we validated several risk factors we know for Parkinson's disease, and we confirmed that certainly age is one of the variables. Pesticide exposure can induce Parkinson's disease or increase the risk of developing Parkinson's disease. And as we all know, coffee consumption could protect from Parkinson's. In addition, we identified one SNP that could offer protection against Parkinson's disease. The results, to be honest, were not so fantastic for us. We were looking for more. Maybe it's a mistake in the design. Maybe it's a mistake in not doing a whole exome sequencing, for example, and the false conception that, okay, we already know a lot of risk SNPs, let's screen them and that's it. So this was a very good lesson for our group that, our environmental architecture is different. Our genetic makeup is totally different. We have to come up with our unique understanding. That's why we continued in our steps. So we are now studying again environment and lifestyle and diet through studying microbiota and Parkinson's disease in Egypt. While studying, we're trying to confirm our our gene study on cell culture. As I mentioned before, sometimes we have this bidirectional movement. So it's not always from the lab to the clinic, but sometimes from the clinic back to the lab. So we identify certain steps. We need to confirm this on cell line or cell culture. That's okay. It's a step of translation, because later on we will move forward to adoption of novel therapies or solutions based on our discoveries. So this was the first project, the first step in ENND. With that in mind, we said, okay, we have to move forward with better understanding of genetics in our population. So we focus in the next project on identifying genes or mutations with familial cases. As I mentioned before, familial cases of Parkinson's disease and most neurodegenerative disease is not considered the majority. However, the value is you can identify with certainty one gene, and with this gene, you could identify one pathway or track that's responsible for the disease. This helps us. This is another project called GeneFINDER, and this is the peak of even. First output was this nice piece of paper published a few years ago. It's not in Parkinson's disease, by the way. It's related to dopa-responsive dystonia, but somehow we identified novel mutation. We are now working on identifying a panel for a gene. I would like to call it Egyptian panel for genes in familial or families with Parkinson's disease. And I guess this would be very helpful. Well, is that all? No. Because while we are doing our analysis from the previous study and this study, we noticed something very weird. We are all analyzing our data against a reference genome built using European ancestry. So it could be very helpful when you analyze data coming up from Germany, from UK, from USA. But when you are analyzing the data coming from Africa, from Egypt, you'll find some mistake. You have some problem. And as usual, starting first step in research, it's the first step. Sometimes it takes you into another different track. And this is what happened with our network, where we drafted the first reference genome for Egyptians. This work was just released a few weeks ago. And with this reference genome, we found out that we need to revisit most of our genetic data published previously. We have to start reanalyze most of GWAS, most of the genetic analysis done before, because simply we were referencing using another reference. It is not our reference. It is a European ancestor reference. Now we have an Egyptian reference we can depend on to impute more data, to analyze accurately many data, not only for neurodegenerative disease, but also for all disease that has certain contribution from genetics. I consider drafting an Egyptian reference genome with, of course, most of colleagues working, one of the greatest achievements for Egyptian network for neurodegenerative disease, and it will be reflected later on all our works and all our achievements, hopefully to come in the next years. So this was a part of genetics we achieved through ENND. And I guess the next step would be joining the IPDGC-Africa, start working together with African colleagues to better understand the genetic backgrounds of disease, especially Parkinson's disease in Africa, based on real African studies to appreciate the genetic and environmental factor in Africa that contribute to the disease. Start doing our homework. Our homework is to understand the determinants of our disease, our determinants for disease in our countries. So I guess joining IPDGC-Africa would be an amazing step for ENND. Okay. The next goal for ENND was biomarkers. As I mentioned previously, biomarkers are very important because you need early diagnostics. And most of the time, we cannot diagnose early because the patient arrives while most of the neurons are lost. The biomarkers are important also because of economic value. So many of the research centers in Europe and the States and other countries are trying to validate fancy imaging techniques, fancy proteomic approaches. That's fine, of course. They're very important. However, when we are talking about hospitals in rural areas in Egypt, and Africa, and other African countries. We are seeking an accessible, low-cost testing methodologies that could identify brain damage and certainly could identify a disease. So we adopted this methodology. The hypothesis here is that simply when there is a brain damage, there will be some release of neuronal proteins that have been damaged into the blood. And this will initiate or irritate the immune system or trigger the immune system to release antibodies. We are detecting those antibodies. So we started this, and this was built on an old methodology. However, our hypothesis here is that if we could have a battery, let's say, of 10 autoantibodies against neuronal proteins, and we could adopt some statistical or analytical method, we could maybe, I'm not sure, but maybe we could reach to certain unique patterns for each disease. So with our preliminary data, we could, for example, for Parkinson's disease, we could differentiate between both conditions using tupulin and tau. In other research, we used the alpha-synuclein autoantibodies. In other research, we used the batteries or a panel of autoantibodies, around 10 autoantibodies. And they showed differentiation between different neurodegenerative diseases. We have two stages. First, to identify or diagnose or confirm that there is brain damage. And this is evident from all our research previously. Next stage, which we are involved in now, is try to come up with idea whether the pattern of elevation and depression of those autoantibodies could be unique to each disorder or not. If we have 10 autoantibodies, could we learn or understand from the patterns of elevation whether this is Parkinson's disease or Alzheimer's disease? The value of this test is the low cost, accessibility. It can be improved to be a simple ELISA test, rapid bit side test similar to pregnancy test and others. Can you imagine this? So we are trying to do this. However, as I mentioned previously, also when you start the first step in research, sometimes it takes you into another pathway or road. So being involved in a multidisciplinary team, we understood that maybe it's not only biochemical biomarkers. So yeah, something. First time we thought about it, we thought it could be crazy idea if we can use login or activities on Facebook as determinants or early predictors of dementia. Is it possible? When I proposed the idea to my colleagues at GBHI, they thought, you're crazy. Very nice, but you're crazy. However, we got funding from Alzheimer's Association, Alzheimer's Society. However, the initial algorithm developed by my colleagues who are experts in artificial intelligence in Egypt and machine learning could be possible. The very early findings shows that we could differentiate between normal and decline in cognition. So who knows? Who knows also with the understanding of a multidisciplinary team or multidisciplinary research that it might not be only one biomarker. It might not be only the biochemical biomarkers in blood. It could be blood biomarkers plus Facebook interaction plus some imaging techniques. They all could build up a system, a paradigm for early prediction of neurodegenerative disease, which I guess could be a game changer in our battle against neurodegeneration and Parkinson's disease. Okay, so we started working with biomarker. We came up with some nice biomarkers. However, the main challenge is they are diagnostic biomarkers, and we need early diagnostic or early predictors. All the time we are trying them on already diagnosed and identified patients. So you cannot validate whether you said that this is a very good biomarker, it's only diagnostic. But if you need to validate, you need to say whether it appears earlier before the development of clinical manifestation or not, you have to start a longitudinal study, and that's what we are doing now. ENND started or launched the first Egyptian longitudinal study of aging with the aim of following up aging population for 10 years from the age of 50 till the age of 60, trying to understand determinants of healthy aging and determinants of aging-related multimorbidities. Al-Sahha, which is the nomenclature for a longitudinal study of Egyptian healthy aging, is the first in Egypt. I guess it's the first in the Middle East. And we are harmonizing with other HRS studies. We are partnering with SHARE, which is the longitudinal study of aging in the European Union. And recently we have listed as one of HRS harmonized studies. Inshallah, hopefully we are start next year here in Egypt. I guess it's the first time Egypt contribute to such magnificent study planning for 10 years. Finally, I like this quote. It always seems impossible until it is done. So usually you feel it's impossible. Could we start genetic work in Parkinson disease in Africa? It's impossible. Could we start a longitudinal study in Africa? It's impossible. Can we identify certain biomarker of neurodegenerative disease? It's impossible. And then they are done. So they are possible. Thank you so much. And I'm open for any questions. Thank you.
Thank you so much, Dr. Salama, for that very interesting talk. I think it's very impressive, the work that you and the consortium of the ENND were able to do over these years. And I think particularly in the African context, I think we need a lot more of these kinds of consortia working together. So as you said, having an Egyptian reference genome or a Nigerian reference genome, etc., I think it would be really powerful for us to better understand Parkinson's or other diseases in the Africa context. Yeah, so we can move on to the Q&A session. So this was, the first question was quite general. Is there a particular reason why, I guess, the consortium chose to focus on Parkinson's disease?
Actually, my PhD was in Parkinson's disease. When I returned back, I found several colleagues who shared my passion and interest in starting on Parkinson's disease. That's why we started working on Parkinson's disease. Certainly, we are expanding now to include other neurodegenerative disorders.
Okay. Yeah, so another question from Sarah. She asks, how can she, as an early career researcher, join the Egyptian network of neurodegenerative diseases?
Yes, of course. Any scientist or clinician or anyone who would like to join us, we are open. The membership is open. Just send me to my email, and sure, we will have the opportunity also to continue or restart our meetings regularly as we did before. So yeah, ENND is open to anyone who might be interested.
Okay, so anyone who's interested, please reach out, get in contact with Dr. Salama. And then she went on to ask, what is your take about looking for a biomarker for diseases as PD and Alzheimer's when there's no curative treatment for them? I always get this feedback when I propose research ideas based on identifying biomarkers.
Yeah. So the issue here is that we don't have a cure or a treatment for an end-stage disease. So we are working on Parkinson's disease after clinical diagnosis. That's the core of developing a biomarker, because if we can diagnose a disease very early in the process of developing a disease before clinical manifestations, we will start the battle much earlier. So we don't know, but I guess this is one of the solutions. Since our neuroprotective agents are not helping curing a disease when most of the neurons are lost, let's try to cure the disease before loss of most of them. So yeah, certainly it will help reshape our battle.
Absolutely. This is quite an interesting question. So this comes from Sharip, and they asked, since we are the result of evolution, what do you think of the evolution of Parkinson's disease related genes?
I have no idea. To be honest, I have no idea.
Yeah, it's a tough one.
Yeah.
Someone was asking again, I think a more general question, have there been any, I guess, interesting or unique findings from the Egyptian Parkinson studies, maybe that you didn't share in the book?
What, I didn't get the question.
Were there any interesting findings outside of what you spoke about during the presentation from the Egyptian Parkinson's disease?
Yeah, very interesting finding. Actually, that most of the published studies shows that smoking is protective against Parkinson's disease. So in our study, no, smoking was not a protective agent. And we started thinking, by the way, why? And yeah, it's a point of debate. And I had a very nice idea, but we couldn't propose or we couldn't prove that maybe because actually the Egyptian cigarettes, also exposed to pesticides. So it could be another risk factor. But honestly, this was very, very interesting. When we started analyzing the data, we were asking ourselves, how come? And most of the studies shows that smoking is protected, but we couldn't find. This was one. Also, there are other genes screened and not published in this, and they were negative. It was amazing how many well-established risk genes worldwide, and we couldn't find in Egyptian. So it's another one. So those are the most important two findings in our study.
Yeah, super interesting. Apparently some people raised their hands, but I don't see. Yeah, if you have a question, could you just type it in the Q&A or in the chat box? That's a lot easier because I can't see if you raise your hand. So if anyone still has questions, just type them in the Q&A. That's easier. So I had a question. I'm not sure if this is known yet, but is there any idea of the prevalence of Parkinson's generally in the Egyptian population and how that compares?
Yeah. Actually, it's a very good question, because when we applied for funding the first time, I think it was around 8 years ago. So we had a conditional approval based on to prove if it is a prevalent disease or not. I mean, Parkinson's disease, especially in Egypt, we have other health issues, health problems. So according to the data we received, yeah, it's much higher than international standards. And in certain places, especially in rural areas, it's around 10 times the international level. So it's a prevalent disease. Yeah.
Okay, that's pretty interesting. Someone is asking for your email address. Maybe you can actually type it in the chat, and then people can copy paste it. We're still on the Zoom. Yeah, the two people with their hands raised. If you can type in your questions, please do so. Otherwise, currently, do you have any collaborations with any other African groups or that are also working on Parkinson's? Is that something that?
Yeah. Now I guess we are all collaborating through the IPDGC Africa, and I guess it's an ideal platform. So we are all joining forces. So we are collaborating with Nigeria, with Ethiopia, with Sudan, and I guess it's a perfect platform to start collaboration between African colleagues interested in one disease.
Yeah. No, I think that will be really interesting because it's always been something I'm quite curious about because there's not as much information coming out of the continent relating to TB. So I think it's going to be exciting. Yeah, so someone also asked, are there efforts to expand the ENND to the Middle East? Are there efforts to expand the ENND to the Middle East? And how do you ensure that the diagnostic criteria are the same throughout the region?
Well, expanding to the Middle East, we are collaborating. But yeah, I guess we are focusing now on Africa for collaboration through IPDGC. However, our longitudinal study, which is launched by ENND, is now extending. We are trying to expand to include Lebanon and Jordan. So we'll have East Mediterranean region. Certainly, we'll be happy to collaborate. What was the rest of the question?
Okay. Yeah, I think the IPDGC definitely provides quite a great for Africans across. Yeah, so I don't see any more questions in the chat. Yeah, so then with that, since there's no more questions, I'd like to thank you again, Dr. Salama, for this really interesting talk. I think a lot of us are looking forward to seeing the next steps in the research that you're doing through the ENND. And we're also looking forward, of course, to the collaborations that will come through from this. So we'll definitely keep posted on.
Certainly. Thank you so much, and thanks to IPDGC and to everyone who attended. Thank you.
Okay. Thank you, everyone, for being here, and we will see you during the next talk. Have a lovely morning, afternoon, or evening, depending where you are located. Yeah, so thanks, everyone, for being here, and yeah.